So with that, I would like to introduce our first speaker, our keynote speaker for this Congress, Dr. Danya Vears. Danya is a senior research fellow and a team leader at the Biomedical and Ethics Group at the Murdoch Children's Research Institute in Australia. She's a social scientist and with a background in genetic counselling, and she's using both empirical and theoretical methods to explore the practical, social, and ethical issues related to genetics and genomics, both in a clinical setting and research settings. She also holds an honorary position at the University of Melbourne, as well as having a guest professor at the Centre for Biomedical Ethics in Leuven, Belgium. And I'm very happy and looking forward to listen to her today talking about examining gene ethics through a genetic counselling lens. So welcome, Dr. Danya Vears.
Thank you. Can you hear me okay? Great. So I'd first like to just thank the organising committee for inviting me to present today. When they gave me the briefing and they said, can you please reflect on how your genetic counselling training has influenced the work that you do? I was like, what am I going to say for 50 minutes? But then I gave it some thought and I realised that, and here's I'm giving you the answer straight away. Actually, genetic counselling really shapes the way I think about the world, and so it shapes the way I do my research a lot. So I've got a lot to say, and I'm going to take up the full 50 minutes for you. So I'm going to just start by telling you a bit about how I got into genetic counselling, and then why I decided not to be a genetic counsellor. No offence. I'm going to talk a little bit about what genethics is in the sense of what I do. I'm going to talk a bit about what genetic counselling skills I remember learning, which was a while ago, but how they influence my research. And I'm going to talk through all the different stages of a research project and just give you some examples about how that works for me. And then I'm going to talk about some of the other ways that I think genetic counselling has made me make a difference in terms of the work that I do. So I first found out about genetic counselling as a profession in my Year 12 biology class. There was one tiny paragraph in the textbook that said what genetic counselling was, and I went, genetics, psychology, two of my favourite things, that seems like the perfect career for me. So I did a degree at La Trobe University in Melbourne, which was a Bachelor of Behavioural Science, and that was because I could do a genetics and psychology double major, so I was like really focused on being a genetic counsellor. And then I remember in my textbook, there was like at least 3 pages about what genetic counselling was by that point. And I was like, okay, this is something that it's actually a thing. And one of the things, I've just pulled this out of the textbook, which I still have, which is strange. But you can see it's from 2000, and there's like a couple of lines there that just say that there are currently 22, this is obviously a US-based textbook, there are currently 22 genetic counselling programs in the United States. Approximately 4 to 7 students are accepted into each program each year, and an average of 130 new counsellors enter the workforce each year. And I just wanted to point that out because think about how much we've grown since then as a profession. I think that's really amazing, and we should reflect on that growth. So I did that degree, and then I waited a while because I realised I was 21 and had no life experience and didn't really feel like I could be a genetic counsellor at that point. So I went and got a research job in epilepsy genetics for a while. And then in 2008, I was one of the first graduating classes of the 2-year masters in Melbourne. So I was one of the guinea pigs. And this is our class. I thought I'd throw in a few photos for you. We were all like looking very young at that point in time. That was a great experience. We did have like a few teething problems, but the course coordinator, Margaret Sahar, was amazing. And we had Jan Hodgson and Lisette Kerno, who were wonderful in terms of coordinating different aspects of the subjects. So I'm very grateful to them. And then you can just see, this is us looking very happy about getting our degrees at the end of that. So I came out of genetic counselling degree thinking, yep, I'm going to be a genetic counsellor. And then some other things happened. Firstly, there weren't very many jobs. So we're talking about 2009, 2010. It wasn't really a big thing in Australia yet, and pretty much all the positions that were available were on maternity leave. I went for a couple of jobs, got a couple of interviews. They said, we want people with experience. And I was like, well, how do you get experience? I'm sure we've all faced that. And then I'd also had some feedback during one of my counselling placements that made me question whether it was really the right thing for me or not. And so I gave that some thought. And then, this gets a bit personal, sorry, but then my mum passed away from breast cancer. And all the jobs were in cancer research, cancer counselling, and I thought, I don't have the capacity to do that right now. So I thought, I'm going to do something different. I'm going to do a PhD. And one of my tutors from my master's course was Professor Lynn Gillam, who is one of the most amazing bioethicists and humans in the world, and she agreed to be my supervisor. So I was like, yep, I'm in, I'm going to do a PhD. So my PhD was looking at ethical issues around carrier testing in children, because I was really passionate about ethical issues around children and that sort of genetic testing area. So I did that for 3.5 years. And then during my last year of my PhD, Jan Hodgson, who I mentioned earlier, said to me, you need to go over to the ESHG and you need to meet people and try and get a job. And I highly recommend that to anyone who's doing a PhD. Go travel, go to conferences, meet people. It's the best way to get work if you decide you want to be in research. So I went to ESHG meeting in 2014. I also went to the COMET conference, which is Communication, Medicine and Ethics, which I don't know if that exists anymore. Met some people there. And one of the people I met was Professor Pascal Bory. And I went and spent a week with his team at the University of Leuven in Belgium. And shortly after that, he said, he Skyped me and he said, so I've got a 1.5 year postdoc position in Belgium if you'll think about moving. And I was like, sure. So I ended up finishing my PhD in 6 months. That's when they were allowed to give you balloons and you had to hand in things in person rather than all online. Finished my PhD and then moved to Belgium in June 2015. And that was an amazing experience. I learned a lot. And it was a beautiful place to live. It was the centre of Europe, so I got to travel a lot, which was great. But I had lots of experience through Pascal Bory because he was a really great supervisor. And one of the cool things I got to do was stay at the Brochet Foundation in Switzerland. And I'm just mentioning that because I think everyone should think about doing that if you want to do some research writing, because they basically sponsor you to stay there on this beautiful location for a month or 2 months and just write. And you meet people from all kinds of walks of life. And I got to go to the WHO building, we got a tour, and one of the people I met was Professor Nina Halliwell, who, if any of you know her, she was the most amazing gene ethicist that I've ever known, and the world is a sadder place for her not being around anymore. But she was a great mentor and friend for me during that time. So then my brother said to me in 2018 that he and his wife were pregnant and I needed to move back to Australia. So I applied for a job and was very fortunate to get a job under Professor Julian Savulescu at the University of Melbourne, and I stayed there for 2 years and then got another job with the same team but through the Murdoch Children's Research Institute, which is where I am today, and then was promoted to team leader in 2021. So that's how I am where I am now. So what's genetics? Well, I had a look on PubMed, and the first time it was mentioned was in 1990, and actually there aren't that many references to it as I was expecting. I thought there'd be a lot more because it's a term that we use quite a lot. But this 2004 paper described it as genetics is the study of the ethical issues that arise out of the science of genetics and the uses of genetic technologies. And that's basically what I do. That's my job in like one sentence. What's interesting is this paper went on to talk about whether genetics is special or not quite a lot. And I'm not going to have that debate with you today because we'd be here for another 2 hours. But I think there are elements of genetics that are a little bit different, and I think it is worthy of its own kind of subspecialty area. So that's my view on things. But the things that I work on predominantly through my postdoc initially when I was in Belgium, I did a lot of work around genomic sequencing technologies, thinking about things like consent. I do a lot of interviews and focus groups with different stakeholders, and so I've done some work with laboratory scientists, looking at how they analyse their data and what they report, particularly to do with things like variants of uncertain significance and incidental findings and secondary findings. And I've done some work with clinicians looking at perspectives on return of results from patients as well. So that was sort of my postdoc time. Since I moved to Australia, I've done a lot of work around rapid genomic sequencing in critically ill children, which is taking off in a lot of places around the world. And there are obviously lots of issues around that time critical period with consent, and we did some offering of secondary findings to these families as well. And I'll talk a little bit about some of that work. And that was with my co-team leader, Chris Gingell, who was leading that grant that we worked on for that project. Since then, Dr. Fiona Lynch, who was my postdoc and who just went and had a baby, we've done a lot of work together on data sharing and data storage. We've also done some work in the last year or so on genomic newborn screening and looking at different perspectives, and I'll talk about some of that data today. And then more recently, I've been looking a little bit in areas around artificial intelligence and using genomics in that space, and particularly to do with automating reanalysis. So I'll talk a little bit about that too. So when I was thinking about what to tell you today, I was thinking about all the different genetic counselling skills and components that I learned during my training. And I haven't labelled any of them here because we're going to label them as we go through, but I want you to think about what all these symbols mean, and some of them are a little bit tricky. But there are lots of things that we learn as genetic counsellors that I think are highly applicable to the research setting and that really shape the way that I do my work. So this first one is a little bit hard. This is client-centred approaches. So the way that my research works and the way that I get funding is I either write myself a grant, like a fellowship, to do a research project, or more commonly, I actually get contracted by another person who's doing a much bigger grant, usually to do with some kind of new technology or some kind of sequencing. And they say, can you please do an ethics stream for us within that grant? And so I'm going to talk to you about a couple of those, but the way what I think I bring to those grants is that I bring that kind of patient, family, public perspective to things, when I guess a lot of the researchers I speak to are very focused on the technology itself. So I think that is a really important thing that the training that I had as a genetic counsellor has helped me bring to these projects. And one example of that was the acute care genomics program that was led by Professor Zornitza Stark through the Victorian Clinical Genetics Service. And that ran from 2018 to 2022. And what they did was they were basically doing this rapid or ultra-rapid genomic sequencing in critically ill children. And these are all the hospitals that participated in that project. So they sequenced 450 infants and children. They did trios first with whole exome sequencing and then moved to whole genome sequencing. For anyone who knows anything about genomic sequencing, which I'm sure a lot of you do, their time to report was less than 3 days, which is phenomenal. And they worked very hard to produce that, and it made a lot of difference to patient care. And they had a diagnostic yield of 53%. So Zornitza Stark wrote that grant, and at the same time as she wrote that, we actually wrote our own grant in my team, where we developed a grant for the Centre for Ethics of Paediatric Genomics, which we call CPIG. And you can see here, this is the structure of the grant, and you can see that at the very top of the purple lines, we're looking at the impact on families. And that was something that I think that was really important, that we were trying to sort of engage with these families and see how they felt about this technology, what was working for them, what wasn't working for them, how they were feeling about the consent process, etc. So that's just one example of one of the grants that we've contributed to. Something we've been working on much more recently is what we call Baby Screen Plus, and that is also led by Professor Zornitza Stark and also Associate Professor Sebastian Lunk at the Victorian Clinical Genetics Service. And what we're doing is we're piloting the feasibility and acceptability of using genomic technologies in newborn screening programs. So we're going to be sequencing, we've actually started recruitment now, which is very exciting. We're going to be sequencing 1,000 newborn babies in Victoria in Australia. And so what I'm doing as part of that grant, because I'm the ethics lead, is trying to engage with the public to see what they think about a lot of these issues and use of this technology. So we're looking at different benefits and concerns that they perceive from the technology. We're looking at implementation issues, things like when and how to offer the testing, which conditions to test for, what kind of consent should be obtained from these families, and importantly, how do we pay for it? And so we've been doing a lot of focus groups that last about an hour and a half. We've also been using a Mentimeter program, which I'm not sure if people have heard of, but it's where we get, so during the focus groups that I'm running, Fiona Lynch was pulling out all the different characteristics that people were talking about, and we're using that to then get people to rank them in order of importance at the end of the session. And that data is going to be used to develop a discrete choice experiment with a much larger population. So that's sort of feeding into how we're developing the program for the newborn screening for the whole of Victoria. So we're learning a lot through that. We interestingly recruited via Facebook ads, which worked quite well in some ways, but we did have a little bit of skewing towards people who were on maternity leave, who had time to talk to me, and who were obviously very interested. And we screened for eligibility and diversity to try and increase the diversity of the people we're speaking to. And I'm just going to just give you a brief rundown of how many people we spoke to for this, because it's quite a large study. We did 15 focus groups with 75 participants in total. And you can see there that we had quite a large age group between 23 and 72 years of age. We had mostly people from metropolitan areas, but we did get some people from rural areas. Most people had children, but some people didn't. Most people who were born in Australia and spoke English at home, but we did try to get some people who had a little bit more, who were a bit more culturally diverse. And then we also got some men, which is hard to do in these studies, but we managed it, which was good. So that's just another example of a program that we've been looking at. This little symbol represents promoting autonomy. And I'm going to talk about how that works in a couple of different ways. So the way I promote autonomy and how that works with my work is that when I'm designing recruitment approaches, particularly with patients, I think it's super important that we remember to allow them opportunities to not participate in the work that we're doing. And so often we recruit through health professionals, and I think we need to have ways that make sure that people feel like that's not going to impact their care. So that's just something to keep in mind when we're doing research, because I know that a lot of people who are doing genetic counselling research will be trying to recruit some of their own participants, and we need to be mindful of that. I also think that the work that I do promotes autonomy in the sense that I try to, with all of the work that I do, allow people to think about allowing other people to have choices. I'm going to explain what I mean by that. But in the newborn screening program work that we've been doing, we felt it was important to ask the participants, who are members of the public, how they felt about parents being allowed to decide what kinds of conditions their children were being screened for. And that's not something that everyone has done. They've often said, you know, what kind of conditions do you think should be screened for? But we've actually said, do you think parents should have a choice? And so a lot of people did feel that parents should have a choice about what conditions are screened for in their baby. That's a tricky thing to do, and obviously a long list of conditions where you can tick boxes is not really appropriate and time-consuming. But we talked about the idea of grouping conditions in terms of things like treatability or untreatability, severity or mild kind of nature of conditions and things like that. So I think that kind of work where we try to allow people to have choices or to experience choices, I think that's really driven by my genetic counselling training as well. So there's another project I've been working on, which is all about reanalyzing genomic data. And so just to give you some background to that, we know that many patients who undergo genomic sequencing don't get a diagnosis straight away. And that depends a lot on what kind of condition they have. But we also know that genomic data is stored and that things change over time. So we're always getting new information, databases are getting more and more variants put in them. So it makes sense that if we look at the data again, we would find more variants. And this is just a very complicated diagram that just shows that reanalysis has lots of different components in it, and we need to remember that. What we do know is that if we waited approximately 2 years after that initial diagnosis, we would get like about a 10 to 15% increase in diagnoses for these patients, which is great, and we need to keep doing this. We also know that there are quite a few guidelines that highly recommend that we do this reanalysis routinely, but it's not happening in a lot of places. So in Australia, from 2018 to 2021, there were 25,000 genomic tests approximately done, and 17,000 of those were unsolved. And so we know that if we just reanalyse that data, we would get more diagnoses. But only about 900 of those have been reanalysed so far, and it's really because it's a time-consuming process. Labs are barely keeping up with the demand of current sequencing, let alone reanalysing old samples. But if we continued at this rate, it would take us 85 years to get through all those samples, which is just not okay, right? So we need to do something a bit differently. There's also issues with the way that the triggers happen for reanalysis at the moment. So there's a lot of inequity because either patients come in with a new set of symptoms, or maybe they're told to come back after 5 years or 2 years by their clinician. Not everyone remembers that. Sometimes they come back in because they want a new treatment and they get reassessed then, but not everyone is getting that opportunity to have a reanalysis done. So wouldn't it be great if we could routinely reanalyse all undiagnosed patients? And we think that automating that process is the answer using AI technologies. Because we could identify new gene associations and we could get more diagnoses for these patients. So I've been doing a program of work with some of my students and also my postdoc where we've talked to a bunch of different stakeholders. So this is meant to be clinicians and laboratory scientists. Not sure why one looks like Jesus, but then we've also talked to members of the public, and we're also doing some work at the moment where we're talking to patients or carers of patients. And so we've got a three-pronged approach here, and we're doing these by focus groups. And so the reason I raise that is because this symbol refers to creating safe spaces for discussion, which I think is super important in a lot of the work that we do in research, particularly in focus group situations. And so one of the things I'm very careful about at the start of all my focus groups is setting ground rules for the discussion. And I've just put, I don't know if you can read that there. Oh, you could, you can, that's good. So I always talk through these at the start, so making sure that they know that I'm expecting them to do talking and want them to be actively engaged. That there aren't any right or wrong answers, that everyone's perspectives are really important and they should feel that they can not necessarily argue but disagree with each other, and that would be okay. To try not to speak over each other, so we'd use a lot of virtual hands in these sessions, and also to respect each other's privacy, because people do share some very personal experiences in these sessions, and we want to make sure they feel comfortable to do that. So that is something that I'm very careful about, and I think is highly influenced by the fact that I have a genetic counselling degree. This symbol refers to being mindful of power imbalances as well. And so that's something that I'm always very careful of when I'm introducing myself and when I'm speaking to people during these focus groups or interviews as well. So the way I frame myself varies quite a lot depending on who I'm speaking to. So if I am talking to genetic counsellors and clinical geneticists, I'm going to tell them I have a genetic counselling degree because I want to be an insider. But if I'm not talking to them, if I'm talking to members of the public or patients, I may not disclose that straight away. I won't hide it, but I want them to feel like I'm more like one of them, because I want people to feel comfortable to talk to me as though we're on the same level. So that's just something to be mindful. And we always talk about in genetic counselling, you know, like dressing appropriately and things like that. That also applies in the research setting. So that's just something I keep in mind. This symbol is for active listening and assessing understanding. But the understanding that I'm talking about here is not for the participants, it's actually my own. I want to make sure that I'm not making assumptions about what they're saying based on my position, my professionalisation as well. So I might have a certain view because of the way I'm trained. So just making sure I'm checking in with them and paraphrasing and things, the same as I would in a genetic counselling session. And empathy is super important in a research setting as well. And it helps me to engage with my participants. I do, as I said to you before, we actually have quite a lot of people sharing some very personal stories, and sometimes they share some very painful things that have happened to them and their families. So it's important to acknowledge those in the way you would in a genetic counselling session, but then I have to draw the line there and avoid doing counselling and not problem solving and not trying to help them, because that's not my role. I'm a researcher. So there is sort of a bit of a tension there, but I think it's something just to be aware of. And this is obviously relevant to all research, not just genetics. This symbol is controversial because it's about non-directiveness, and I feel like that's a term that's bandied around a lot and is misused a lot. I think it's a very helpful term to use in some settings, but we need to make sure that it's not abused. I think when I'm thinking about non-directiveness in the research setting, I'm thinking about making sure my questions are open-ended and not leading in any way, because I don't want people to necessarily say what I want them to say. I want them to feel free to talk to me about what's important to them. But also there is an important area where you would be presenting different ideas to them that they may not have thought of themselves, and seeing what they think of that too. And so one example is with the newborn screening programs, when we were talking about the payment models, I allowed people to sort of free talk and just say what kinds of payment models they thought would be appropriate. But then I also raised some other ideas and said, what do you think of this and what do you think of this, and get feedback. So that's more of that sort of shared model of conversation rather than just letting people go off on their own by themselves. And this is about education. And one of the things that can be a little bit tricky in research is when participants are asking me questions about what we're talking about. And so I think that's a fine line to walk on, because obviously I want them to talk to me about what they think is important. But if they've got the wrong idea about something, there has to be some kind of fact checking as well, because it might influence the discussions. So when I'm talking about, when I'm thinking about how to answer a question that people ask me, I think to myself, is the answer integral to their response? And if it isn't, then I kind of throw it back at them. And I say things like, why does that particular aspect matter to you? Or would that make a difference and why? And then it gets them thinking, and it gives me an understanding of what they mean and why it's interesting to them. And I might answer them afterwards because I don't want to be like aversive, but I think that it does give you a bit more of a rich sort of data set when you sort of explore that in a bit more detail. And in terms of other education, with the focus groups I've been doing recently, I've been doing a lot of small short videos, sort of between about 3 and 5 minutes prior to them coming to the focus group. And I found that really helpful because it gives people a little bit of priming about the conversation we're going to be having. Sometimes if it's members of the public and it's quite a tricky topic, like things like reanalysis or using AI in genomics, that can be a lot to sort of go through at the start of a focus group for the first time. But I found that they have found the videos very helpful. And it's amazing the amount of times people go, oh, as I heard on the video. And I'm like, I wonder if they realise it's me, that's my voice as well, that they're listening to. But yeah, I found that really worked quite well in terms of an educational component for these discussions. Still on interviewing and focus groups, I think there is a really important role for questioning and challenging ideas. And I know that there are elements of that in genetic counselling as well, but I think there's a trick to knowing when to dig deeper in these sorts of discussions. One of the questions that is often asked is what kind of findings do people want to receive from different types of tests? We know that people always say, well not always, but a lot of people say, I want everything. Now, the trick is that you need to ask, well, what happens if you can't have everything? And allowing people to make choices. And sometimes you need to push people a little bit and challenge them on that, because I think that that's when you start figuring out what it is that is actually of value to people, rather than just saying, yeah, sure, you can have everything. Similarly, encouraging deliberation is something that I found really helpful as a part of focus groups that I've been running. And this ties into that same issue with things like prioritisation. So with the newborn screening program, we've been doing a little bit of trying to get people to sort of say, well, if you have to prioritise, what would you choose and what would be more important to you? And that's something that the discrete choice experiments will be exploring in a lot more detail as well. And this is just an example. We actually did some polls at the start of some of the focus groups with the newborn screening project, and one of the questions we asked was should a newborn screening program that includes genomics be run any differently from standard newborn screening programs? And pre-discussion, so before we even talked, so we'd had the briefing, we've done the housekeeping and everything, they'd watched the video, but before we had any discussion as a group, 18 of the 39 in this subset of the sample said yes, they should be run differently. 8 said no, and 12 said not sure. Post-discussion, you can see that the number of people who said yes, things should be run differently increased a lot, and that was usually to do with the consent process. So to me, that shows that people, when they had a chance to deliberate and discuss as a group, they understood the complexities a lot more about what a consent process involves and how genomics might be important to do things a little bit differently in that setting. So I think that really shows that it's helpful. So moving on to data analysis, and one of the methods of analysis that I use the most, because I think it's the most appropriate in a genetics setting a lot of the time, is inductive content analysis. And that's a qualitative method for analysing texts, and those texts can be interviews or focus groups, or it can be other data that already exists, like documents. And the goal is to produce an overall summary of multiple texts. And the key features of inductive content analysis are that it's inductive, obviously, but that means that the categories are developed from the data rather than predetermined, like a list that you're just looking for. And it's also iterative, and that means that you're coding repeatedly. So you go through a couple of transcripts, you go back and compare with each other all the different transcripts. And this is a method I used in my PhD quite a lot, and my PhD supervisor and I found during my PhD that there was actually not that much information about how to do it well. So we wrote a document, a paper about it, which was a guide for beginning qualitative researchers. And I've been giving it to my students for a while and they find it really helpful, so hope if people want to give that to their students, it might be helpful as well. We actually just also wrote another piece of correspondence for the Journal of Genetic Counselling, trying to sort of unpack some of the confusion around that as a methodology. And I think what's important to think about is when it's useful. So it's useful because it's inductive, it's when there's not much known about the topic, and that you can use thematic analysis as well, but that's another potential option. I think what's more important is what are the intended outcomes of the research. And if you're trying to do something that's really practical and you want to develop recommendations or try and improve practice, then it's actually much more useful than thematic analysis because it's not so heavily tied to a theoretical framework. And now I'm going to turn to dissemination, and this is my favourite symbol. It's promoting empowerment. And there's been a lot of work that's been done in this space, and there are sort of 5 main components to promoting empowerment that I've listed there. And I just want to acknowledge that Marion McAllister has really driven a lot of this work and used a lot of it in the genetic counselling outcome scale, and that's been pivotal in this space. So in terms of the research setting, obviously in genetic counselling we try to promote empowerment a lot, but I think there are elements of that in the research setting as well. So we know that there can be a therapeutic effect from discussion, and I think that really helps some people who participate and can be a driver for participation. I feel like my role is really to give people a voice, and I take that very seriously in my research because they're entrusting me with their stories, their experiences, their views, their perspectives, and knowing that those views are going to be given a voice and might actually help develop things like policy or change practice, I think really helps people feel like they're making a difference and might have hope for a better future. And one example of that is where in this reanalysis program that we were doing, where we presented a model of potential automated reanalysis to our participants, and in this setting I'm talking about the clinical people and the laboratory scientists. And we said, here's the model we're thinking about. What do you think about that? What are your concerns? What would work? What wouldn't work for you? And that's not something that we do on a large scale all the time. And so this was a really great study for them, and they found it really helpful to have a chance to talk through their concerns. And I just want to mention some of those to you. So I'm actually going to present you a tiny bit of data today. So I'm going to talk about the concerns that they had. And some of them were about inability to recontact the family after a reanalysis happened. So as this clinical geneticist said, they may have moved, and then the issue of if you have a result, what's the legal obligation of disclosing that result if you can't get hold of that patient? They also had concerns about where the boundaries of moral responsibilities to disclose results were. So how hard did they have to try? So as this clinical geneticist said, well, if I haven't been able to reach this patient, is it my responsibility to contact their GP or attempt to contact their GP, or are there other treating team? Like, how far do you go and what's reasonable for you to absolve that responsibility? So there's a lot of concern about holding on to results and not being able to give them back to the patients. They had concerns about risks to the clinical relationship as well. And so as this clinical geneticist said, none of us wanted to come out later on in the piece that this result was available and there may have been effort to contact that was then aborted. You know, why didn't you try harder, sort of thing. That could be pretty rapport destroying as well. And there was worries about risks to institutional reputations or public trust. So as this pathologist said, it only requires 1 or 2 cases to have really big payouts, financially and reputational damage, and that actually ruins it for everyone else. You just have so many examples medically, misinformation on social media, and how that can run away so quickly, that particularly in what we do and the implications of that to healthcare from the information that we provide clinicians and patients, it doesn't take very much for us to lose a great deal of trust in the public and actually cause a great deal of reputational and financial damage if we don't do it in the proper way from the get-go. They had concerns about lacking funding for follow-up of these families as well. So as this clinical geneticist said, certainly from a funding point of view, we then need a new referral, so it's difficult for a service to cold canvas call a patient after many years. And they were worried about insurance implications for these patients as well. So as this person said, thinking about insurance implications and things like that as well, I guess our knowledge is growing about the spectrum of the disorders and how that might be linked to other things, and we might not have prepared them for that information. And some patients might have got insurance before testing and then cancelled it thinking that we didn't find anything or not renewed it, and then we give them another result. So that was really interesting because some of those concerns were not what we were thinking about in terms of the ethical implications of this data. We weren't thinking that they were the main concerns. We were more worried about the implementation side of things. So I think it was good that we got that information for that study. And then there's also educational aspects to do with dissemination of the information as well. So we're very careful to write summaries for our participants before we actually publish the research. We try and get summaries back to them so that they feel like they're involved in the process and they have a good sense of what other people said, and people are very interested in that. Education also extends to supervision. And I supervise a bunch of different students, not all of these at the same time, just saying, but at all different levels. And this is just some examples of some of the projects that I've supervised at PhD level and Master of Genetic Counselling students, and we have a Master of Genomics and Health course in Melbourne as well. And I also supervise people at Master of Bioethics in Belgium. And I think this is really important because I'm very passionate about training junior researchers. And I know that supervision is super important in terms of our genetic counselling training as well. So I think that really instilled in me that necessity to nurture junior researchers and to make sure that we give them a future, because academia is not easy. I think also education applies to how we target our audiences, and I'm very particular about the journals that I try to publish in as well. So this is just an example from the Centre for Ethics of Paediatric Genomics grant, which is finished now, so I can show you that there's a bunch of papers. This is not all of them, but the ones with stars are ones that are in non-bioethics journals. And that's kind of unusual in some of the bioethics spheres. A lot of people do try and publish in bioethics spaces, but I feel like the work that I do, I want it to make a big difference. And so I feel like it's important that the people that I'm speaking to through the papers, the voices that I'm having heard through the papers that I write, get to the right audiences. So they need to go to clinicians, they need to go to lab scientists, they need to go to policymakers. Ethicists are not necessarily the right audience for that. And up the very top, you see where it says a question of ethics. That's actually some blog work that we did as well, so we could make it more accessible to a public audience too. And I think that's also an important part of education in terms of the work we do. But most importantly, I want my work to be to develop practical recommendations and to sort of help inform practice. And that doesn't happen just through papers. So I'm also very involved in a lot of different committees, and I'm just going to talk through some of those for you. So I'm still very involved in genetic counselling in some ways because I'm a member of the University of Melbourne Genetic Counselling Course Research Committee. And on that committee I do a lot of marking of theses, as well as trying to help develop practices for future for the subject and trying to just refine practices. And I find that's really great because it helps keep me connected to the genetic counselling community as well. Through the Qualitative Research Community of Practice at the Melbourne Children's Campus, I'm on the steering committee of that, and we've been, over the last, I think 5 years I've been on that committee, we've been really trying to make sure that the quality of the qualitative research that's being done on the campus has been improving vastly, and that's really important because there are a lot of clinicians, so genetic counsellors and other types of clinicians that use these kind of methodologies, but they don't necessarily have the training for it, and we've been doing a lot of training in that space, so that's really important. I'm also part of the Generation Victoria working groups for the ethics and governance and the genetic ethics lead. And so Gen V is actually a program that's being run through the Murdoch Children's Research Institute, where they're recruiting as many babies as possible over a 2-year period, and they're nearly finished. And they're going to be taking some biosamples and a lot of data from those kids and following them through to try and sort of help understand some of the problems that happen in children from both a health perspective, a mental health perspective, and all that kind of stuff as well. So that's a really great initiative, and I'm very pleased to have been involved in that, and we're hoping to launch genomics in that space as well. So watch out for that. Other policy work I do nationally is that I'm the co-chair of the Education, Ethics and Social Issues Committee of the Human Genetic Society of Australasia, and that's something I feel really passionate about. It's a great work. We do a lot of work on that committee to try and improve practices, and we do a lot of policy work as well. So As part of that role, I've been leading the development of a bunch of different position statements that have been approved by the Human Genetics Society of Australasia that are highly relevant to genetic counselling. So we've done some work around predictive and pre-symptomatic testing in adults and children, carrier testing in adults and children for recessive conditions, and most recently the work that we've been doing on our grant for the rapid genomic sequencing. We've actually developed a position statement for that, and that was published in the European Journal of Human Genetics about 2 weeks ago. So that was exciting. But that just shows that the work we're doing on that committee is actually pretty groundbreaking in terms of having an impact both in Australia but also internationally as well. And as I said, that's really driven by my desire to make a difference, which I think is highly tied into my genetic counselling training. At an international level, I'm on the Global Alliance for Genomics and Health, and I've been on a bunch of different task teams of that. And with that, I've also developed some policy documents. So we did some position statements around implications of data sharing in paediatric genomics, and also some in newborn screening and use of genomics in that space as well. And I potentially stupidly at one point agreed to lead a big systematic review of all stakeholder perspectives on individual return of results from genomic research, which was way more work than I was anticipating. It was about 221 papers. But that was really important work because it actually helped develop a policy from that. And so the 2021 policy on clinically actionable genomic research results was developed based on that data. So again, trying to make a difference in the world. And then finally, I'm also part of the Scientific Program Committee for the European Society of Human Genetics, and that's a real honour because it's really exciting helping develop the content for conferences, as I'm sure the organisers here feel. It's really exciting to be able to provide a space for people to present their work, to showcase the best research that's happening in that space. So I'm going to give a little plug here for ESHG in 2024, which is in Berlin, one of the coolest cities in the world. And I can say from personal experience, the content of the program is looking great, if I do say so myself. And for those who might have attended ESHG last year, the DJs, which is me and Celine Lewis, which we call ourselves the Chromatones, we've been booked again. So hopefully you'll come to the party and have a dance with us. So I've got plenty of time for questions, which is good, but just to summarise, as I said to you at the beginning, genetic counselling is really the lens that I look at the world through, and so therefore it influences everything about the research that I do. It guides the questions that I ask. It makes sure that I include patient and family perspectives. It influences who I engage with, so I try to get a range of different stakeholders so we get all the different perspectives heard to provide voices for everybody. I think it's made me much more aware of the impact I have on my research in general, so I think about my interactions with my participants more than I would have without that. It makes me more empathetic, it makes me more motivated to disseminate the research that I do more meaningfully, so it makes a difference. And it really drives me to nurture junior researchers and to help provide a commitment to improve practices through policies and committees and all that kind of stuff. And that's why I'm always so busy, but I feel like it's really important work. And I feel like if you're going to do research and you're using taxpayer money, then you need to make the most of that. And I feel like that genetic counselling has made me see the importance of that work as well. So there's a lot of people that have been involved in the research that I've just presented to you today, all the different studies, but I'm particularly thankful for all the patients, carers, members of the public and health professionals and clinicians, etc., that have been part of the research that I've done. And I'm happy to take questions. Thank you.
Danya, that was great. And while we give everyone a moment to think of what questions you want to ask her, I just want to say that I feel like you really highlighted so wonderfully some of the struggles that we have as people trained as genetic counselors who are thinking about making that leap into research. We worry we're going to give up part of that genetic counseling side. And you just highlighted so nicely how there's this great overlap in skills, but also these challenges and tensions that come up. And I feel like people of every level of research could learn something from what you said. So I want to thank you for an excellent talk. Do we have any questions in the audience right now? Things you'd like to ask Danya, either about her personal career or perhaps some of the research she's doing? Yes, please, Annie.
Do you need a microphone?
Do you want? Is there anybody else?
Oh,
Yes.
you're on.
Yeah.
Oh, you've got one.
I've
Okay.
got a mic here. No? So I was thinking of when you were having a focus group, especially for the baby screen programs, and in your thing I saw that there were more females than males. So do you sometimes take the male parent and the female parent's opinions separately and see whether they...
Okay. So in that study, we were actually just talking to members of the public, so they weren't actually participants. But we are going to be doing some interviews with participants from the baby screen study as well. And we were actually just having this conversation the other day and saying that we should be, although the birth parent is the person who will be included in the research and they have to sign the consent form, we also said we should be offering the other parent the opportunity to have a say as well. And I have done that in other studies as well, where I've said, you know, normally through snowball kind of sampling techniques to say to the mother, or who's usually the person who is involved in the study, does your partner want to have a say in this as well? I think doing those interviews separately is better, but I have actually had couples say they want to do them together, and that's, you know, tricky because they talk over each other and one's always more dominant than the other. But I think you have to, including the perspective is better than not including the perspective if you get the opportunity to.
Yeah, because that's something a genetic counselor does, no? Takes the couple together and...
Yeah. I mean, I have heard of people who say, you know, do you want to talk about things separately depending on what the situation is in a counselling setting as well. I think if you have, I mean, obviously in a research setting, I have more time to be able to offer that. In a clinical setting, you don't necessarily have the time to give them an extra appointment. That's tricky.
We have a question over here, somewhere. Yeah, in the front. Hang on, there's
Just
a
wait a
microphone
sec.
coming.
You'll get a microphone.
Thank you. It was a very nice talk. I was wondering, I have a question about the reanalysis of the samples. Do the parents give consent in the first instance, or do you ask the parents to reanalyze, or you reanalyze the samples and then you have the information and then...
That is a great question, because I was going to present some data about that and then I thought I wouldn't have time, but it turned out I would have had time. So that's a really tricky situation, because obviously we've got all these samples that we don't necessarily have consent for reanalysis for. It depends. In Australia, each different state has been, up until recently, had their own consent forms, and so they all target it at different levels. And obviously, we don't know what the health professional is saying alongside the consent form as well to explain what reanalysis might or might not happen. I think most of the time, the forms will just say, we may want to look at the data again in the future in light of new information or something like that. So it's very vague. And so particularly when we're talking about reanalyzing using potentially AI, I think that's a really tricky question. And so I did ask participants who are members of the public how they would feel about reconsent not happening, whether they thought that we could just assume that it was part of the original test. And people were generally not okay with that. People felt like we needed to get reconsent for the reanalysis. And health professionals feel the same way about that too. They feel like if we don't get reconsent, then we're going to have situations where people don't want that information, and then we're stuck with a result that people don't want. And particularly if it's been a long time since they've actually had the sample taken, like we were just talking about from 2018, but there are obviously samples from well before that we would want to reanalyse as well. So I think it's the problem is there's just so much of a backlog of people we would need to get reconsent from. So I think we don't have the answer to that yet. But yeah, great question.
Hi, Danya. That was a really great talk. I really enjoyed it. Further to the reanalysis study you're doing, I'm really interested to hear what comes from that, because I was involved in a pretty large-scale reanalysis program where we retested people who had testing for BRCA1 and 2 sometimes many years ago, and we managed to get a waiver of consent to retest many clinical samples. And it was a big job, and then we recontacted people to give them the result. And it was a lot of work in some cases to recontact people, and sometimes people had died and we returned the result to a next of kin. But we interviewed some of the people we recontacted, and they were overwhelmingly really positive about that having been done, and I think the clinical relationship that they had actually supported a lot of their attitudes to it, rather than it being harmful that we hadn't reconsented them. They actually kind of saw it as within scope of the original testing, because it was, we were looking for causes of hereditary breast ovarian cancer. So yeah, I'd be really keen to chat to you about what we learned from speaking to the participants, if you have a chance at something during
that
the
would be
conference.
great. And I actually had heard about your study because of this project I've been doing and been meaning to talk to you about that. I think that, I personally think that a waiver of consent would be fine. The problem is that there are always going to be situations where, for example, I mean, a lot of the samples we're talking about are not breast cancer, we're talking about rare disease patients, and a lot of them are kids. So there's like extra complexities with that. So some of those might be that, say, for example, that child has died and the parents get a result and they're really distressed about that. Sometimes it might be that they're actually, the parents are now pregnant with another child, and they might be really upset that they didn't find out before they got pregnant. So there's things like that that are tricky. And then you're also going to get to situations where those children have become adults, and then they haven't even given consent for their sample to be tested themselves, and they may not know what happened. And so how do you negotiate that? So I think that there are lots of complexities associated with that. But I do think, I think that it's valid to think about that waiver of consent option. And yeah, I'd like to, in my mind, I really think it's part of the original test. But I guess I have to talk to the clinicians, and they don't necessarily feel that that's appropriate, and we don't necessarily have proper consent from these families.
Other questions? I see one back there. We can pass the mic back, maybe.
Hi, I work at the Newborns Screening Project in the UK, and I'd be interested, maybe a question to take up over coffee, but in terms of the consent, how do you balance ensuring that people have enough information to have given meaningful consent versus overloading people and entering into a sort of paternalistic dialogue about how much they need to know, and kind of placing too much emphasis on an educational model? Have you wrestled with that problem, and how have you addressed it? As I said, you've got 15 seconds, so it might be one for coffee. But
yeah, it is a really tricky question. I was only sort of semi-involved with that, with the development of the information. They're doing two prongs of recruitment, and one is in person with someone actually talking to them, and there's also an app that we're testing out. The app is not getting a lot of traction right now, and so we're just trying to figure out how to make that work better. But I think ultimately, with something like newborn screening, when it's at that kind of scale, we are, although it might have to be mentioned by someone like who's a health professional, I don't think that we actually have the capacity to do that at a major level without using things like apps or videos or something like that online. So I think we need to think about that in a bit more detail.
thank you.
You do have more than 15 seconds if you want to elaborate on that.
No.
Do we have other questions? I didn't realize everyone could see that timer counting down. Yes, over here with the microphone.
So we're now in question time.
Yes.
I was
That's
running
right. That was
early.
just for the talk.
Hi, Danya. Hi, it's Chris Jacobs.
Hi, Chris.
Hi there. I'm just going to come back to the thing that you so beautifully talked about the role looking through the genetic counselor lens when you're doing research. And I think one of the things you talked about very well is that temptation to counsel when you're doing focus groups, and to pull, and how hard that is to pull back, and how important it is to be reflexive when you're doing that. And I wondered if you could talk a bit about how inductive content analysis, where the role of reflexivity is in inductive content analysis, because I don't know.
That's a really good question. So I guess, I think it's the same as any type of qualitative analysis, that you need to be really aware about what you're bringing to the research you do. And so being mindful of any perspectives that you already hold about that particular topic that you're researching. I think that's why it's important to have teamwork. So I'm a very firm believer in co-coding. So that's, for anyone who doesn't know, that's where you might have a primary person doing the coding of the research using something like inductive content analysis, but you have someone else who checks at least a proportion of those, and you talk through where you might have had discrepancies in terms of how you've coded the data. And I think that's where you can find out that sometimes you're over-interpreting what people have been saying, which is one of the most common things I find with students is that when I'm co-coding their data, they might be like coding it as something like autonomy, and I'm like, A, that's a very complex topic. They didn't actually say that, and you need to code with inductive content analysis quite close to the data. But B, they might just be talking about, something they've mentioned having a choice or something like that. And I think it's really important to have that checking mechanism, particularly with students, to make sure that they have someone else who can just kind of question them and challenge them on some of the things that they're coding, because I do see that students particularly, but I'm sure I have done it myself, is that you have an idea about what people are saying and then you run with that idea in the coding. And so, yeah, does that answer your question?
Yeah, it does. Yeah, I think you made the point, I don't know if you can hear me. You made the point there, because if something like reflexive thematic analysis is part of the methodology, and I wonder, in inductive content analysis, I don't know that it is, apart from being a qualitative researcher and knowing that's an important aspect, I think. So it's just maybe reminding students or others that are using it that that's an important aspect.
Yeah, I always get worried when I hear that there hasn't been co-coding done, particularly with student projects. But I think I do think you have a responsibility as a qualitative researcher to be reflexive in your work, and so I think any time you're doing a project, you need to sort of sit back and think about what you're bringing to it as well.
Other questions? Yes, one more in the back.
Thanks so much for your presentation. I was struck by what you were talking about when we want our research to make a difference. Do you have any tips or tricks for doing like actual policy engaged research that results in policy change and impact at that level?
I guess my strategy has just been to join committees, but I guess that's not necessary. You can't join hundreds of committees. I think it's often about who you know. And so I think trying to connect with people who are on committees is probably a valuable way of doing that too. I think that, for me it's critical to the work that I do, but not everyone feels that way, but I think that, if you're going to do the work, you should actually make the most of it. So yeah, I think trying to engage with people who actually are on committees, if you're not, is probably a good way of doing it. That's all I can think of right now, but we can always chat more in the break if I come up with anything else.
Danya, I have one last question, if you'll indulge me a little bit.
You've got 30 seconds.
Well, it's a somewhat personal question, but I think in the spirit of this meeting, where we're here to celebrate and encourage genetic counseling research, I want to ask you a two-part question. What is the best advice you ever got as you started your research career? Mm-hmm. And part two, what advice do you wish you had gotten?
You've given me no time to think about it.
I know, sorry.
Okay, so I think the best advice, particularly for my PhD, was about containment, and that's something I try and talk to my PhD students about, as well as my PhD supervisor, Lynn Gillum, said to me, this is not the Nobel Prize you're trying to win with this research. This is just you learning to be an independent researcher, so it's a training ground. And so don't put too much pressure on yourself to try and solve all the problems in the field. What I wish I had learned... self-care. I think, it's very easy to push yourself in academia. There's a lot of expectations about what you have to produce to get through, and it is a lot of pressure. And so I think, I'm constantly being told to take care of myself, and I think that we need to try and create a culture where that is acceptable, and not even acceptable, but encouraged. Thanks.
All right. Well, Danya, you are making a difference in the world, as you said, and I want everybody to thank Danya for her great talk.
Thank you.